E aí pessoal, tudo bem? Como estão? Gostando da RBCC? Bom, a RBCC tá um evento top, em virtual, palestra boa, tema importante. Tá de parabéns, comissão organizadora? Show de bola. Bom, mas eu venho aqui para convidá-los a conhecer a Biostock. Pessoal, a Biostock é o maior shopping online referente ao mercado científico. Lá vocês vão encontrar equipamentos e consumíveis que com certeza vão atender a necessidade de vocês. E ó, eu já estive aí no lugar de vocês. Esperar o orçamento, esperar informações de nota fiscal é muito chato e demora muito tempo. Então, ó, rapidamente na Biostock, por ter vários lojistas, você consegue obter em cliques os seus 3 orçamentos e digo mais, inserir na nota fiscal as informações referentes ao seu processo. Ó, facilidade assim na mão de vocês, no celular de vocês. Então, pessoal, acessem biostock.com.br, conheçam a nossa plataforma e se vocês procurarem algum produto que não estiver em nenhuma das lojas, por favor, chame Bia, nossa assistente virtual, e escreva: Bia, procurei por tal produto e não encontrei. Nós iremos fazer um esforço máximo para atendê-lo. Bom, eu sou Bruno Gamba, Head de Negócios da Biostock e desejo para vocês um excelente evento. Até a próxima, pessoal. Abraços.
Eu não consegui te ouvir.
Hi, everybody. Can you listen me? Oh, great, great. Thank you. Sorry, everybody. I will start again. Hello. So, boa tarde a todos e a todas. Espero que vocês estejam me ouvindo agora. Eu tive um problema com o áudio. Então, estamos de novo aqui, né, para mais um seminário, para mais um webinar do RBCC. E, como todas as vezes, a gente está, né, tendo esses encontros, discutindo citogenética e mantendo, né, essa nossa motivação e esse nosso engajamento, esperando ansiosos pelo congresso no próximo ano. A gente tem o prazer de ter um convidado hoje, o Jiri Macas, que vai dar uma palestra em inglês, e por conta disso eu vou retomar a apresentação agora em inglês. So, again, welcome, Jiri and Ludmila. So, it's a pleasure to have you here today to give your talk for the Brazilian cytogenetic community. Today, Ludmila will help me with the moderation of this talk. And I would like to introduce her. Ludmila Oliveira had her master's and PhD in the Federal University of Lavras with the supervision of Dr. Giovanna. And she also did a postdoc in the lab of Jiming Jiang. Now she's postdoc in the lab of Jiri, and she will help during this talk. So, Ludmila, welcome, and please introduce the speaker to us.
Okay, thank you, Andrea, for the kind introduction. Thank you for the organizers to give me the opportunity to be here. I will today introduce you Dr. Jiri Macas, or simply Jirka, which is much easier name for us Brazilians to pronounce. So I first met him in 2015 when I came to spend 1 month in his lab. At the time, I was encouraged to do that by my former master and PhD supervisor, Dr. Giovanna Torres. And I'm really thankful for that opportunity, because I got to know him better in the lab, and also the other way around, like he got to know me a little bit better. And I think it helped for him to give me the opportunity in 2018 to join his lab as a postdoc. So I have been here for almost 3 years now, and then I have the honor to be part of his group since then. So enough about me, let's then talk about him. So Dr. Jiri Macas is a biologist. He did his master in plant physiology in the Masaryk University in Brno, and his PhD in plant molecular biology in the University of South Bohemia, Český Budějovice, which he concluded in 95. During his PhD in 91 and 92, he worked in the laboratory of Dr. Ingo Schubert in the IPK, in the big city of Gattersleben in Germany. He was also trained as a fellowship of the European Molecular Biology Organization in Italy and as a postdoctoral fellow in the University of Arizona in Tucson, USA. Along his career, Jirka published about 140 papers, besides some books, chapters, and very well-deserved awards he received. He has been cited more than 5,000 times by now. As many of you know, Jirka and his group have a strong reputation for a beautiful and very effective combination of bioinformatic and cytogenetic tools to study mainly repetitive DNA. So his research is focused on the development of bioinformatic tools for identification and characterization of repetitive sequences and the analysis of those repeats in plant genomes. Also studying the molecular structure and function of plant centromeres, including the beautiful metapolycentric and holocentric chromosomes in plant genomes, and the origin and evolution of supernumerary B and sex chromosomes, and many times also focusing on LTR retrotransposons. Since 95, he started as a research scientist and principal investigator at the Institute of Plant Molecular Biology in the Biology Center of the Czech Academy of Science in České Budějovice in Czech Republic. And I could not introduce Jirka and do not mention, of course, the Repeat Explorer, which he created together with Pavel Newman and Petr Novak, also from our group. So the Repeat Explorer pipeline was designed to identify and characterize repetitive DNA elements in the next generation sequencing data from plant and animal genomes. I have to highlight here that together with the platform, which is free, they also offer the use of a public server to run the analysis and a workshop to teach people how to use it. Inclusive, the last year workshop was online. So it's really a complete package that I think is safe to say that it changed the reality and gave so many more possibilities to cytogeneticists around the world, like to find very nice, interesting probes for their works. Especially the last year during the pandemic, it was very useful to everybody. So thanks, Jirka, for creating this platform. Thanks for being here talking to us today. And I will give you the floor.
Yeah, thanks, Ludmila, for this detailed introduction. I really feel very old now after the list of all these achievements. But yeah, it's my great pleasure to be with you and to present in your online meeting. Of course, I would much prefer to be in Brazil with a glass of caipirinha in my hand and talk to you in person. But nevertheless, this is a situation. So today I will talk about very peculiar chromosomes in Pisum. I would share the screen. Okay, I hope you can see it.
Yes, we can.
Yes.
Thanks. So I would like to give you a sort of overview of our, let's say, last 9 or 10 years of work on this very interesting type of chromosomes or centromere. So first, I would like to introduce these metapolycentric or expanded centromeres of Pisum. Then I will take you to the journey through technologies and time, showing you how we actually gradually looked at different features of these chromosomes, and also which was also determined by the possibilities or technical advancements that gradually, become available for like different kinds of analysis. And then I will focus on our recent work on this topic, when we try to actually assemble complete centromeres in Pisum and provide you with some first insights we get from these efforts. So you are well aware that the centromere types actually come into basic forms. So one is monocentric chromosomes, where the centromere is localized into a single region. It appears as a primary constriction in metaphase chromosomes, and as generally it is the active centromere is marked by the presence of a specific variant of histone H3, which we call CEN-H3 in plants. It's called CEN-A in human and in some animals. And in most plants, as well as in other organisms, it marks the place of the active centromere. So contrary to the monocentric chromosomes, there exists a holocentric organization of centromeric chromatin, where it is dispersed along the whole chromosome length. And in plants, most of the species have a monocentric organization, but there are groups of plants that are holocentric, and it's well demonstrated that the transition from monocentric to holocentric chromosomes occurred multiple times independently during the evolution of higher plants, while we don't have many cases or almost any of the reverse transition. So it is not known, and this is one thing we would definitely like to elucidate, what actually triggers this transition and how it is facilitated, because we believe that if we would elucidate the mechanism of this transition, we would learn much more about the general principles of determination of centromeres and the inheritance of centromeres and so on. So this is why we were really excited when about 9 or 10 years ago we have discovered this type of centromeres in Pisum, which is a plant which was used by Gregor Mernut for his experiment, which is very important for us because he was also Czech, as we are, and he worked in Brno, where actually I studied. So we are big ties to this man, and so it was, as I say, it was a discovery based on the observations that really, because we were using P as a model to study repetitive DNA, and we noticed that really the centromeres in this species are unusually extended. And when we looked at the localization of some repeats, they gave strange patterns. So we really started to be very interested in this feature and started to study in more detail. And so the first thing, of course, was that we wanted to see the distribution of the CEN-H3 chromatin, and we were really surprised to see that really it doesn't show features typical for either monocentric or holocentric chromosomes, but it was something in between. So along these extended constrictions, we could see multiple domains of CEN-H3 chromatin, and later when we looked at other species like Latyrus, which is closely related to Pisum, we could see the same feature, but in Latyrus it was even like more extended on some chromosomes. So you can see here that it's almost like 1/3 of the chromosome actually is represented by these extended centromeres and multiple independent domains of centromeric chromatin. So we studied this phenomenon in a, let's say, wider selection of species from the legume tribe Fabae, which includes, in addition to Pisum and Latyrus, the genera of Vicia and also Lens. And we did a thorough phylogenetic analysis, and we also analyzed the presence of different CEN-H3 or the presence of and the sequences of CEN-H3 coding genes. And of course, we looked at the chromosomes with the SNH3 labeling. And what was interesting was that we found that the occurrence of those extended centromeres, which we started to call metapolycentric, correlated with the presence of two relatively diverged copies of SNH3 genes, while this sister lineage of Vicia and Lens, it contains just one of these genes and it has a normal monocentric chromosomes. We did a phylogenetic analysis and found that actually there was a duplication of SNH3 genes at the base of this group during the phylogeny, and then one of these copies was lost in the lineage going to Vicia and Lens, while both were retained and probably diversified in Latirus and Pisum. And it is interesting to note that like SNH3 genes typically are present in only a single copy in most plants. So even in polyploids, after the polyploidization, there is a reduction of the number of copies to one, usually. So of course, coincidence doesn't mean causality. So we don't really know how it if it could be some sort of a mechanism or the conflict that actually triggered this centromere expansion. This is one of those things we are interested in, but it's relatively complicated. Anyway, so we started to ask questions such as what is the sequence organization of this specific type of centromeres, if it corresponds to di or multicentric chromosomes, maybe that they originated by some sort of a fusion, or is it rather similar to holocentromeres? Also, what is interesting, how the 3D arrangement of SNH3 chromatin emerged, because you see that SNH3 again is always located at the edge of the constriction, of course, because this is how it needs to be for the segregation during mitosis. And of course, what is the origin of this metacentric organization? And we were aware that it will not be an easy task, because Pisum has a relatively large genome of more than 4 gigabases per haploid genome, and we estimated sizes of these constrictions into related species in Pisum sativum and fulvum, and could see that there's anything between like 50 up to 130 megabases long. It was estimated just by measurements of the fluorescence of chromatin or on chromosomes. So it's a rough estimation, but relatively good. So we knew that if we want to answer these questions, we really need to integrate data from like various methodologies. We will not be able to study it using a single methodology. And at that time, it really was out of question that we could even think about like assembling such a very large and complex, because it's repeat rich sequence. So we were using methods that look at these chromosomes from different perspectives. From the large scale, it would be like mostly cytogenetics methods, and also at the same time or in parallel, we are trying to do from the very tiny scale, looking at the sequences. And this is what I would like to briefly present here at the beginning. So talking about the cytogenetic level, here you can see this is also quite interesting feature of these chromosomes, how the SNH3 or the centromeric chromatin looks during the cell cycle. And what is really interesting is that these spots or these multiple domains, which can be seen on a condensed metaphase chromosome, actually get even more condensed in interphase, and they just make one spot per chromosome. So here you have 14 SNH3 domains that actually represent these spots fused together. And during the cell division, this gets dispersed and reorganized to form this structure on the metaphase chromosomes. And we can see using electron microscopy that these domains can be distinguished even in this very high resolution on the electron microscope. This is just the constriction with 3 SNH3 domains. And we also checked how these domains interact with tubulin in the mitotic spindle, and we could see that all of them actually are connected to the spindle, so all of them work like small centromeres, which work in accord. Then we also started to look more closely to SNH3 genes. As I said, that there are 2 of them, and they are divergent on their N termini. So we designed a specific antibody for each of these proteins and colocalized them and used high-resolution thin microscopy to visualize them on chromosomes. And you can see that they are colocalized. So there is not a case that there would be one SNH3 domain made just one of these variants. They are always present both in all domains. And we investigated also epigenetic modifications, and were relatively surprised to see that they actually resemble those of holocentric chromosomes. For example, different kinds of phosphorylation, they show very similar distribution as can be seen on holocentric chromosomes. I mean the phosphorylations which are present in these constrictions. There is very interesting modification, which is this phosphorylation of threonine 120 in H2A, which actually seems to make like a boundaries around the SNH3 domains, and it actually speculated that it can somehow mark these domains in the chromatin. So going to the other side of this scheme, again at this time, we started to do some Illumina sequencing because it was a technology that was, the mostly available. And as Ludmila already mentioned, we developed this pipeline repeat explorer for analyzing repeats using shotgun Illumina reads. So we of course applied to P genome, and thanks to that, we could characterize overall composition and copy numbers or proportions of various repeats in this genome. But of course, it didn't tell us which of them are centromeric repeats. So for that, we had to do ChIP-seq. So we did chromatin immunoprecipitation using SNH3 antibody. So in this method, the chromatin is fragmented to like nucleosome-sized or denucleosome-sized particles, and then those that interact with the SNH3 antibody due to the presence of SNH3 are immunoprecipitated and isolated, and the DNA is sequenced. In parallel, we always have a control of total chromatin, and then we used the output of Repeat Explorer, which produces clusters, which are sort of collections of sequences sorted based on their origin. So each cluster represents a different repetitive sequence. So we use them as a reference to map these ChIP-seq and input reads, and we calculated a ratio. And of course, those clusters where the ratio was where there was an enrichment of SNH3 immunoprecipitated reads compared to the control, those were those that potentially are associated with centromeric chromatin. So you see that there is quite a lot of dots here. So there were relatively many such repeats identified, and we verified these with FISH and the combination with immunostaining of SNH3. And we were again surprised, not for the first time in Pisum, that there was really a huge diversity of centromeric satellites as suggested by this analysis, but also that on a single chromosome we were finding in individual SNH3 spots or SNH3 domains, we were finding totally different repeats. And this is something which is completely different from the situation in human or in Arabidopsis, where there is a single centromeric satellite that makes one centromere, and it's present in centromeres of all chromosomes. So here there was a really huge diversity of the satellites, not only in the number and the types of sequences, but also in their combinations and distributions on chromosomes. So it was quite frequent that like each domain on one chromosome was made by a different satellite, but in some chromosomes all domains were made of the same one, and then you can see these all different patterns which we could see on the 7 chromosomes of Pisum. So it shows really that these centromeres are probably highly dynamic in their repeat contents. The other analysis we did was that we wanted to actually have some markers for centromeres, some cytogenetic markers to study them in more detail, and maybe also to find homologous centromeres in related species to follow the fate of these regions in different species. And for that, we started to develop single copy fish markers based on EST markers from the Pisum genetic map, which was published by Taech in 2015. And here on this graph, you can see the markers, because they generated really thousands of EST-based markers and high-density genetic maps. And here you can see these markers sorted according to their position on chromosome 6, which is here in centimorgans. And because the centromere is actually the region where the meiotic recombination is suppressed, so all markers that come from this region actually have like the same position on the genetic map. So from that, we selected some of these sequences, which were like anything between 1.5 to 3 kb long only. So you really needed to optimize the fish to visualize such small targets. And you can see the example of one such a probe again, which localizes in the constriction in the centromere of centromere 6. So we generated some number of these probes, and later on, when the first version of the Pisum assembly became available, we participated in these efforts. So we also used our cytogenetic markers and also these EST probes to verify or to check the quality of the assembly in the centromeric regions. And unfortunately, as expected, because this assembly was made with mostly with short reads, we found out that also the assembly generally is very good for genes or low copy regions. It is partially depleted for high copy repeats, and especially there are many gaps and misplacements of contigs in the centromeric regions, and the centromeres are generally poorly represented. So it was clear that we really cannot use this assembly for really thorough study of the centromere structure and some features we were interested in. So thankfully, during this time, there were already some other technologies available, and we directly jumped to nanopore sequencing, because we were aware that we really need something which will provide us very long reads that can span those repeat-rich regions like satellite repeats or high density of transposons and so on, to get this long-range information for a reliable centromere scaffolding and assembly. So we optimize the protocol of DNA isolation and nanopore sequencing for Pisum with the aim really to get as long reads as possible, not so much to get a high throughput, but to get a long reads. And this is an example of the graph of the read length distribution from several sequencing experiments using this MinION sequencer, where we generated, really relatively high number of reads. And you can see that we really have a very good representation of reads that go over 100 kb. and we had even some reads that were up to like 800 kb. So then when you are lucky and such read contains almost a whole array of the satellites, so it's much, much easier to assemble it compared to some other technologies. So these are some statistics from the sequencing. We, of course, used almost all these reads for doing a new version of the assembly in collaboration with Jonathan Kreplak and Judith Burstein from France, from INRA, who were also leading the first assembly. But what we really were mostly interested were these fractions of the longest reads, like over 160 kb, because even in this fraction, we still had a relatively good coverage, almost 5x genome coverage. And you can see that like half of this data was, for example, in reads that were longer than 200 kb. So we decided, because that's another problem, that really the assembly of the centromeric regions is extremely challenging, and there could be many errors, in the placement and the structure of satellite repeats and so on. So we said that we will not trust any automatic assembler, and then we will do it like in a controlled way, because we really needed something we can trust and we can use it for benchmarking our other attempts of assembly. So we did a sort of a semi-manual scaffolding of the whole centromere of one selected chromosome. Yeah, so and we selected this centromere of chromosome 6, not only because it's the biggest one, but because it has also a good configuration of the satellite repeats. So there is this these red dots. This is a satellite which we call FABTR10, and it's present on many. It's just like the most abundant centromeric satellite. It's present on many chromosomes, but there are also 3 satellite repeats, these 3, which are specific for this single centromere. And we wanted to use it also as some sort of a cytogenetic markers for the assembly and so on. So what we did was that now we are going back to these genetic markers which were localized by fish, and we knew that their position within the centromere. So we used one of them and selected reads that contained the sequence, and we always were focusing of verifying everything by getting multiple reads, or for making each region being covered by multiple reads, so we would know that we don't have any artificial reads and so on. And we were using preferentially this fraction of the longest reads to extend this scaffold into both directions, and again, always verifying the overlaps and so on. So at the end, we got the whole 170 megabase centromere scaffolded this way, and we had it anchored with those single copy markers. So this first, the original one was here, and the other 2 markers actually are localized already within the arms, or at the border of the constriction and the arms. And this whole region had 8 gaps, but otherwise it was like continuous. And of course, because we had a lot of data from fish mapping about the position of satellite repeats, so we correlated our scaffold with a fish map, and you can see that these are all different satellite repeats present within the centromere, and this is how they are distributed in the assembly. So from this, you can also see that the gaps were always within some big cluster of the satellite DNA, which we were not able to assemble. But some of them, like for example, this satellite, we were able to go through because there was some variation, some structural variation. And so this array has like something like 2 megabases. Yeah. Okay, so still we wanted to verify everything by some other methods. So we were using the satellite probes on fish with the Pakitin chromosomes, which are more extended that metaphase, and we were verifying the position and also orientation of these different regions. So finally, we got what we wanted, almost gapless assembly of the whole centromere of one chromosomes. And so we directly started to do some analysis, and this is still going on. So what I will present is how we got so far. So the first thing we could check, of course, is how it correlates with fish on the metaphase chromosome and where the constriction actually starts. So here it is marked by the positions of one of these satellite repeats, FAPTR10. So you can see that its present more or less corresponds to the beginning and end of the constriction. So we could correlate it to these regions of the map or of the scaffold. And we also mapped all genetic markers. And again, here is a plot of their position within the assembly compared to their position on the genetic map. So here you can see that the distance is increasing. Here is a region with suppressed meiotic recombination. And again, here it starts to to change the position on the genetic map. So here we can see that this region with suppressed meiotic recombination is actually much bigger than the primary constriction, so it extends to the chromosome arms, which is also observed in other plant species. And these are some preliminary results where we mapped genes and different types of repeats within to this region. So we could see that really gene and dispersed repeat densities are relatively similar within this centromeric region or within the constriction compared to the chromosome arms. Yes, especially if you look at the LTR retrotransposons, copia and gypsy, you don't see much differences except for positions of the big arrays of satellite repeats, where of course there couldn't be any retrotransposons. What was interesting to see is this type of TY3 gypsy element, which we call CHDCR, and it's actually this lineage of CRM elements which are targeted to centromeres in many plant species. And it has a relatively small copy number in Pisum genome in the range of like, I don't know, 300 or 400 repeats per genome only. But you can see that really here also it is more clustered in the centromeric region, and I will come to back later on. So what we wanted to do as one of the basic analysis was again to find the position of the centromeric chromatin. But for that, we couldn't use our old ChIP-seq data because we didn't have a sufficient number of reads. So we actually ran the new experiments, and so we used antibodies discriminating these two variants of the gene. And we did 2 repetitions for each experiment, and we really used high coverage ChIP-seq. So we had something like 200 million Illumina reads per each repetition. So it would allow us to do this mapping in a high resolution. And this is what we got. So here with different colors are different variants of SNH3 genes, SNH3-1 and SNH3-2, and these are always the 2 repetitions. So you can see that these domains of enriched chip to input values are distributed through the whole centromere, and they correlate with the position of some satellite repeats. But also you should notice that not all of them. Yeah. So for example, this one is free of SNH3, the same for this one, the same for this one, the same for this one. Again, both variants of SNH3 gene colocalize, and it's confirming our previous observations from the high resolution microscopy. As I said, CNH3 is mostly located on arrays of specific satellite repeats, but some peaks are broader, and there are also peaks that are present at satellite-free regions. For example, this one. Yeah, so we don't have any satellite here. So it's not 100% correlated with the position of satellites. And if you look again on the distribution of this centromeric LTR retrotransposons, we can see that not all, but many of them really, again, are positioned within the CNH3 domain. So it really implies, because there were like hypotheses that maybe they are transposing everywhere and they only can survive in the centromeres because it has lower rates of recombination and removal of these elements. But these results rather support the other hypothesis that they really specifically target centromeric chromatin because, and it's also supported by the fact that they integrase has an extra domain, and it is supposed that this domain actually somehow interacts with the centromeric chromatin during the insertion. So this seems to be another proof of this hypothesis. And yeah, so then we were correlating what we could see in the assembly and on this enrichment map or the position of CNH3 chromatin and what we see in fish. And we are basically interested how this actually turns into this arrangement during the metaphase. And you know that there are some quite different models about let's say, arrangement of chromatin within the centromeres. Some of them suppose that the chromatin is arranged in such helices or loops, while some others suggest that the DNA is rather arranged there in some sheets, or it runs like, from across along the centromeres. So it's also this recent work of Kubalova et al. So when we try to correlate our data, so we could say that really our data rather support these models with some turns and loops of chromatin, which goes from the middle of the centromere of from this region to the edge or to the border of the centromere and back, because otherwise we couldn't really explain it. Because if there would be any the arrangement of chromatins that actually runs along the centromere, so all these domains which are at the periphery would have to be located very in one group, within one part of this assembly, and this is not the case. They are distributed along the whole one. So it's much more plausible that the arrangement is something like that. But we would like to study this in more detail using more probes, and especially using chromosome painting probes that should really elucidate this much better. And the last thing, I will just go back to this interesting observation about this tight condensation of chromatin, of centromeric chromatin in the interface. And we actually were tracing those different satellites, those which are associated with CNH3 chromatin and which are on the periphery, in contrary to those which are not associated, and they are located always, in this inner region of the centromere. So we are tracking them during the interface, and we could really see that, doing multicolor fish, is that what is associated with the with a CNH3 and is located at the periphery, all of these satellites always get condensed into one spot during the interface. So it correlates or supports what we see with CNH3, while the others actually get spread, get highly decondensed, and are present in the long distance from this centromeric chromatin. So really the CNH3 chromatin makes a difference and determines the behavior of during the interface and during the cell division, and it differs really quite a lot from the remaining chromatin which is present in the constriction. So this is what we have up to now. So to summarize it, I would just say that we showed that near complete assembly of such a giant centromere, which actually has a size the same as the whole Arabidopsis genome, can be achieved only by a combination of cytogenetic methods and ultra-long sequencing, ultra-long read sequencing. The mapping of SNH3 chipsec reads confirmed the multi-domain structure of centromeric chromatin in Pisum, and both SNH3 variants colocalized. SNH3 domains are distributed along the whole length of the assembled centromeric sequence, and SNH3 domain differ from the surrounding chromatin, as I said, in metaphase and also during the interphase. There is a lot of work we want to do. One is to include PacBio hi-fi reads to assemble all these gaps which are in the satellite arrays, because these reads are better for this purpose, and there are specific assemblers for this task. And we already have the reads, and we did the assembly, and I didn't have time to make slides, but we already have some positive results in that. And of course, we would like to use our experience with assembling this one centromere to assemble all remaining centromeres and compare their features. And we would like to map also the other epigenetic marks. And again, we already have the reads from chipsec with the antibody against this phosphorylation of threonine 120. This is the one which makes these nice surrounding patterns surrounding the SNH3. And I really, if this talk would be tomorrow, I probably could present them, but unfortunately, I had to do it today. And we would like to design painting probes for studying the centromere architecture. And most importantly, we would like to answer the question of how the centromere evolved. And so we would like to identify the orthologous regions in related species in Pisum, in Latyrus, and Vicia, and see what was actually going on with this region. If it was just spreading the centromeric chromatin, or if it was extending the centromere due to the accumulation of some repeats and so on. Okay, so let me thank my collaborators, people from my lab, Pavel, who was in the project from the very beginning, and Iva and Laura, who did most of the fish experiments. Andrea is an expert for the isolation of really good DNA for nanopore sequencing and also running chipsec. Peter does a lot of bioinformatic work. And our colleague from France, who are leading the whole genome assembly in Pisum, and Andreas Huben and Veit Schubert for their help in high-resolution microscopy. And thank you for your attention.
Thank you very much, Jiri. Wonderful, wonderful work, and so much done, and so many interesting things to do still.
So much things to do, yeah.
But it's wonderful when you have so many new results and open up so many new questions, and this result about the centromeric CRM and the targeting, and you have very good evidence for that. It's really very nice, really very nice. How you can explore these multiple centromeres to dissect the influence of the centromeric chromatin to the border, and it's very good system. Very nice. So we will have we are open for questions. So please, if you want to send questions in the chat, me and Ludmila, we will ask them for Jiri. I would like to start with the first question, just to warm up. Actually, I have 3 questions, but I will ask them later on as well. So the first one, I would like to have a bit more detail because of this evolution of the centromere structure. How is the variation regarding size and number of centromeric clusters if you compare within the species and among closely related species, how frequently or this structure, this metapolycentric structure varies, or is it very stable within the species?
Well, it's not. Well, it's stable for particular chromosome, yeah. So we can see it, but there are differences between chromosomes within the species, and like we have, let's say for example, three main domains which we can mostly see on particular chromosome, and on other we mostly see four. But it's really hard to tell because it depends also on the way how you prepare the chromosome spreads and on the condensation and so on. So sometimes some of these domains fuse, and this is one thing we would really like also to investigate, because you could see that on the distribution of these peaks of chipsec enrichment, so they are not really totally regularly spaced. Yeah, this was one thing we expected maybe, but they are not. So it looks like whatever is associated with NH3 chromatin, it's dragged to this periphery of the constriction during the chromosome condensation, because it needs to be there to connect to the kinetochore, and the rest is somehow folded, in between. And so, yeah, to answer your question, not within one species, not all constrictions are the same, and not all of them has the same number of these major NH3 spots. Yeah, in Pisum, sativum, it's not so evident like in other species. So, for example, in this wild species, Pisum fulvum, we sometimes see that more like a line along the centromeres, along the constrictions. And in some wild Latyrus species, we could see that there is a really big difference in the size of the constriction, and some constrictions seems to be really small, while others are bigger. So there is no force that would probably unify them across one genome, yeah, which is also sort of surprising. And yeah, so but we would really like to know why is that, if it's like some stochastic process, just depending when the next chunk of NH3 chromatin is somehow transposed somewhere else and then it stays there, and why is that? Yeah, so of course the question how to target, or the way how to target this, is also to look at different kinetochore proteins, yeah, because it could be that there is some difference or some defect in some chaperones or some other proteins that can determine whether NH3 chromatin is being formed or how it is transmitted to the progeny and so on. So yeah.
So we have a question in the chat from Andre Vanzella. Mm-hmm. He's asking, how is the participation of centromeric retrotransposons and Auger elements in the pericentromeres of these metapolycentric chromosomes? Mm-hmm.
Well, Augers, they really don't show any specific distribution within the constriction. So they don't really show any like enrichment or depletion. And these centromeric elements, this is what I was showing on the plot. So they like, also when we do fish, we see them like concentrated in the constrictions, but there are some spots, or there are some signals which are around the constrictions. So this could be maybe old insertions from the time when the position of the centromere was a little bit different, and they remain there. But there are generally not many of them, as I said, like hundreds per genome. And we would like to see this for like all chromosomes to do some generalization, because if we could see just one centromere, yeah. Mm-hmm. So I hope I answered the question.
Yes. The other question I also wanted to ask was about the satellite diversification, because it's amazing how many different satellites you have in the centromeres, and and it's your also new result regarding the satellites that some are specifically enriched in CNH3 domains, and some are not related at all, just interspersing there somehow. And I wanted to know whether this, if you have checked for the homogenization or structure organization of the satellite, which are directly involved in the centromeric domain, in comparison to those that are not taking part with the interaction of CNH3. Do they evolve quicker, or are they more conserved? Or have you checked the satellite monomere and comparing them in this regard?
Well, it's on the list, but but the thing is that we really want to first make a really good assembly of the, if possible, of the complete arrays. Yeah, and for that we really need to do this hi-fi pack bioread assembly. And but from what I know, because when we were scaffolding that, so of course I was looking at these arrays and so on. So I can say that, like, in many cases, we can see the development of higher order repeats within the centromeric satellite, which is actually something similar we see in other species with CNH3 associated satellite repeats. Yeah. So, but of course, again, we would need to look at the different centromeres also and different satellites, and then to make a synthesis of that. But it will be definitely one thing we want to ask. But you know, in Pisum, it's complicated by the fact that the same satellite can be centromeric on one chromosome and non-centromeric on other chromosomes, and they could be almost, the sequences could be almost the same. Yeah, and we have one locus of one specific satellite in Pisum sativum, which is CNH3 associated, and it's almost at the same position on Pisum fulvum in the related species, but it's not associated with CNH3 chromatin. Yeah, because we have data from this. We don't have assembly, anything like that, but we have cytogenetic data from that, and we know that this is the same cluster because it has a specific mutations which we can distinguish in fish even. Yeah, so we can localize it on fish specifically on both species, and in one species it's associated with CNH3, in other one not, because the centromere is a little bit shorter there. So one conclusion from that is that definitely the primary sequence of these satellites plays no role in its association with CNH3. You know, you cannot speculate about anything like centromere drive, you know, interaction of the sequence of satellites with the CNH3, because there are 13 different satellites which are associated with CNH3, and they are really different, you know, in monomer size, composition, everything. So there is something else going on, and we would like to know what it is, you know, how it happens that, like, really particular cluster or particular array in the centromere of one chromosome has an H3 chromatin, and it doesn't have it in another chromosome. We also have this. Yeah. So what determines this? You know.
This is a very interesting question. And another one I would like to ask myself or comment about it is about this the condensation process of the centromeres. Like, it's very interesting that it seems to do or to go the other way, or the opposite way than the rest of the chromosome, right? Like, you have in interphase the centromeres more condensed, and then it goes like less condensed. So this is very, very interesting, like very unique, I think. So I would like to ask if you would like to go deeper on investigating this and how it works, how it happens.
Yeah. Well, maybe you will have some idea how to do it. No, I'm just kidding. No, definitely, but yeah, it's just too many things we could do, and we cannot at the same time. But this is why I was showing it. It's really striking. Yeah, we didn't expect to see anything like that. That like in the interface, if you just localize CNH3, you have 14 spots. So you would say, okay, that's just normal. You know, more normal species with a normal, chromosomes with one centromeric domain, because you have 14 domains in the interface. And then, when it goes to the mitosis, it starts to disperse it and then partially condense it back, but not entirely. And it remains like this. So it could have something to do with the phosphorylations, because these different phosphorylations, really change the chromatin structure and can act in that. So we know that this H2A phosphorylation is present around these NH3 spots in the interface, but exactly how this works, of course, we have no idea, because I'm not aware about the similar example, from some other species. So yeah, it's yeah, as I say, it's so many different interesting things that emerge there, that simply have to select just something to do.
Yeah.
And there are also another couple of questions. I just following this specific one from Ludmila, and before asking the actual next question, just curious if you see during interphase at any particular stage, or if you cannot tell about specific stage, but in a couple of nuclei that they decondense and then like end of S phase, or just if you see during interphase also a decondensation time for that.
We did not investigate this in detail, really. And this is one thing which I plan to do using chromosome painting probes, if we will be able to develop them. And I would like to have actually a probe that would paint the whole centromere or the constriction, or maybe some parts of it, and then the adjacent regions of the arms, and see it really in a high resolution 3D during the different stages of the cell cycle. Maybe we could even flow sort nuclei, based on the amount of contents of the DNA to do it. And this should really tell us or show us the dynamics of this decondensation and condensation, how it interacts, and so on. So that would be really interesting to do so. The question is if we will be able to have these probes working, because we already actually designed some and they are being synthesized, but the density of oligos is relatively low in these repeat-rich regions, because you need specific sequences. So we will see how it works. I would be happy if we could really do this painting experiment, because it should tell us a lot. And what we could do then is to use the same probe for related species to see what it paints there, how it correlates with the position of centromere in another Pisum species, for example.
Okay. I just wanted to ask the question. The next question was from Danielle. How has any evidence of the emergence of new satellite DNA detected from transposable elements in the pericentromeres of these metapolycentric chromosomes? If you know the origin of these satellite DNAs.
We don't have this evidence for Pisum, but actually in Latyrus there are many satellite repeats that emerge from like a subrepeat, tandem subrepeats, which are present in some types of gypsy elements. And interestingly, they sometimes paint, if you use a probe for these repeats, it totally paints the whole constriction of Latyrus on one chromosome only, for example. Yeah. So again, this is something strange going on there, because it's being this tandem repeat, like 3, 4, 5 copies, is present in many elements of this LTR retrotransposon, and it's dispersed in the genome. And only within these constrictions, but this is not within the NH3 chromatin, this is within the whole constriction, is this amplified as a satellite. Yeah. So, but this is not case for Pisum. It's for Latyrus. So we published a paper about that, I think last year or 2 years ago. So if you are interested, you can find it out. But yeah, so in this case, it looks like maybe the constriction due to the lack of meiotic recombination, maybe it's really like a safe haven for those amplified satellite repeats, that it really is, they are not removed so frequently from there. But yeah, okay. So in Pisum we don't have such evidence.
Yeah, we have just a comment actually complementing this question from Daniel. From the comment is from André Vanzella again, saying that the question is interesting because some satellite DNA could emerge from this UTR region of Auger that occupy pericentromeres.
Yeah. So that's actually this case in Latirus. Yeah.
Mm-hmm.
So it could be like it's not. I think it's the same lineage of the transposons.
Mm-hmm. Yeah, we have many comments of people like saying thanks for your great speech, like from Ana Cristina Brasileiro Vidal, people saying that the fantastic approach, amazing, Wellington Clarindo, Vanzella also saying that the talk was really nice.
Thank you.
Yeah, I don't know if Andrea has more
Yes,
questions.
there was also some comments from Claudio Montenegro, fantastic work, and Adriana Ludwig, beautiful work, and so on. I would have one last question, if you still have the patience. It's just connected to the questions from the pericentromeric heterochromatin, and I was very surprised that you see the suppression of recombination in a region that is larger than the centromere, which is already very extended, but you didn't find any particular repeat or anything that is present in this or enriched in this pericentromeric region that would somehow lead to specific different chromatin domain, or that would explain the suppression of recombination. So I was curious if you have any thoughts about how this region of the, let's say, at least this suppressed chromatin that is adjacent to the centromeres is being defined, or is just a question of, well, it didn't stop exactly at the centromere, so it just went a bit further without any repeat, or whether it has to do with the size of the chromosome and any physical problem of recombination around.
Yeah, well, what I think is that the position of the primary constriction, from what we can see, is just determined by the position of the first and the last spot of the CNH3 chromatin. Yeah. So then there are correlating this phosphorylation and so on. It's like restricted really mostly to this constriction. Yeah. So and then the other part of the chromosome really doesn't look any different. There are satellite repeats on some chromosomes which are close, which are actually within this zone. Yeah, but I couldn't say that there is any enrichment of like the occurrence of satellite repeats. Yeah, so they are then more concentrated on the termini of chromosomes, which is also quite common. And yeah, so I don't know how exactly this is determined, but it is really, I think, more common phenomenon, because I think, for example, in barley, this recombination suppression goes like to the middle of the arms of the chromosomes. It's very far from the centromere. It's actually a big problem for breeding, because then you have many markers in the tight linkage, and people are thinking how to disrupt this. And but I don't know if anybody was really successful in doing so. Yeah. So yeah. So we don't see any like cytogenetic correlation of that. Yeah. And of course, we also don't know how it is on other chromosomes. But I would suppose that it will be like that as well. Because even like I remember this classical work from Jimming's lab about rice centromeres. So there also there was a zone where there was a CNH3 like domains, and then this non-recombining zone was actually much bigger. It's extended much further from that. So it seems to be a general phenomenon, but yeah, so, but I don't have any like hypothesis like about the mechanism how this is facilitated, because it doesn't correlate, for example, with epigenetic patterns. Yeah.
Yeah, because for small genomes, you would have at least, I would imagine you have a pericentromere which is heterochromatic, rich in repeats. So this would maybe give a chromatin condensation that would disturb recombination and access of other factors. But in this case, you don't have any special environment epigenetically or from repeat point of view. What then determine where recombination is free to go, and this region close to the centromere, whether it's small or large, like in barley. Now, what defines these limits? If it's not epigenetics or
but we actually haven't looked really on meiotic chromosomes. What matters in this case? Yeah. So
yeah, definitely.
That's another thing. Yeah.
Yes, very, very. So, thank you very much. I don't know if Ludmila wants to say anything.
I was just wondering if you could tell us a little bit of the history of Repeat Explorer. I mean, I know it's not really the data related to the data you presented here today, but I believe many people used the Repeat Explorer in Brazil. I know many groups using it. So I just wanted to take advantage that you are already here, and maybe you can tell us really shortly, like, how the idea arose, and like if you firstly intended to do something to use more internally in your group, and then you decided to.
Yeah, it was totally selfish, it wasn't for the humankind. No, it was because we were investigating repeats for some time, in the past, and using, as I mentioned before, using like a classical laboratory technique. So we had a robot doing colonies picking and making DNA libraries, and we searched these libraries to identify satellite or to identify repetitive sequences, because at that time it was little known about the diversity of repeats in different plant genomes. There were just a few model species which were selected actually because they had almost no repeats. Yeah, so Arabidopsis is not a good model to study repetitive DNA. So we are looking at the bigger genomes and so on. And this was all extremely laborious. And then we, identified or characterized some repeat. And then we said, okay, so this took us like 3 months or half a year, and it is 3% of the genome. And we don't know what is the rest, and we don't know what is other species. Yeah, so we were like frustrated with that. And then actually there was a time when the first like next generation sequencing technology became available. It was 454 sequencing. I think it was like 2007 or something like that. And so I said, okay, so because we had also before trying to use microarrays, but for microarrays you need to know probe sequences. We didn't know what repeats are there. Yeah. And so we said, okay, in any case we have to try. So we were actually the first among the two labs which were the first using 454 data for repeat analysis. And the first attempts were very primitive. So we just used some software which was developed for analysis of cDNA reads. And it was just putting reads together based on their similarities. And we said, okay, so we will just arrange them like that and quantify them and so on. But it had many problems because repeats are not transcripts. Yeah? In repeats, you have a huge variation, and one repeat is within another. So if you do this sort of primitive clustering, you got one huge cluster that contained all repeats, and it was like very difficult to deal with. And so we were thinking, okay, this is a good way. We need to do some sorting of these reads into some sort of groups that would represent different repeat families, and then maybe we can investigate them. And so then also Peter came to the lab, and he's a great bioinformatician, and he knows a lot of statistics. And so we were thinking how to do it, and we came with this idea of using the graphs for representing the similarities between the reads, and then there was already a background developed for analyzing these graphs, not because of the sequence analysis, but also for other things. Yeah. So, like our first paper about graph-based clustering is, for example, cited in some papers about the water distribution network somewhere. I don't understand it, yeah, but I'm just saying that it's like the similar principles can be used in different applications. So then we started to explore this, and then we realized this is how fantastic this is, because it not only allows us to do this clustering and quantification of repeats, but if we really start to visualize these graphs, it will tell us a lot about the biology of this element, because we see the variation of the sequences and a lot of things, you know, so we can read it from the graphs. So this is actually how it started. So we were doing this type of like exploratory work, and because we had the data and we had the interest and we knew, you know, we are bioinformaticians who were original biologists, which is actually much better because we knew what we wanted and we knew biological questions, yeah, which is quite different from the case when you have an informatician and then you need to explain to him the biological problem, yeah. And so we were just designing it for our needs, yeah, and testing. And so there were circles, you know, we were always developing something, tested it on our data, see if it works, and so on. So it was a lot of programs. It was extremely hard to run them. And so this is how Repeat Explorer pipeline actually originated, that we said, okay, people are interested in using them and they mostly can't because it's very difficult. So we need to wrap it up, connect it, and make it available in some way that people can use it. So this is how this web-based version actually started.
Thank you. Thank you a lot.
Yes, thank you very much again for the time. It was a great pleasure to have you.
Yeah, it was my pleasure.
And just before we end, I would like to thank Ludmila also for the company and for the great presentation you gave to Jiri. And I would like to announce that the next meeting will be still this month, end of October 27th. And Dr. Stefan Pufla from the Research Fellow at Procter & Gamble will talk about validation of the 3D human skin-based micronucleus assay. So I hope to see you soon again. And thank you for your attention. And thank you again for our speaker. And see you next time.
Okay, bye.
Bye-bye.
Bye-bye.